this video is about DNA replication. And that's just the process of making new DNA before we make new cells. So, reasons that we would need to make new cells would be for growth, healing, to replace the dead cells, or in prokaryotes like bacteria and archaea for binary fission or asexual reproduction. So here you can see we have a parent cell and its chromosomes here, and that DNA must replicate so that when the chromosomes pull apart and the cytoplasm pinches apart, we can have two daughter cells that are genetically identical to the original parent cell. One word to describe DNA replication is that it is semiconservative. That means that part of the original parent strand is conserved or stays around. So here's our DNA double helix. This is it if I unwound it and laid it flat with our nitrogenous bases pairing in the middle. And so during DNA replication, these strands break apart from each other. New nitrogenous bases, complementary bases, are added, and now you have two new strands that are genetically identical to the parent strand, and they have half of the parent strand in them. Before we go deeper into DNA replication, we should understand the structure of nucleotides. Nucleotides are made of three pieces. They're made of a phosphate group, a deoxyribose sugar, and some sort of nitrogenous base. In this top nucleotide, I have a purine. And the two purines are adenine and guanine, or A and G, as you may have heard it. The U in purine rhymes with two, so U and two reminds me that there are two rings in a purine nitrogenous base. Conversely, nucleotides with one ring are called pyrimidines. So pyrimidine like pyramid with a Y. And so those are thymine and cytosine, which also have Y's in them. So that's how I remember that cytosine and thymine are pyrimidines. And those only have one ring. But what makes the structure of the nucleotide extra important when it comes to replication is knowing which number of carbon we are looking at. So in this deoxyribose sugar, it has 5 carbons. And in chemistry, they label carbons from like clockwise. So this is an oxygen. So this is the 1' carbon, and the nitrogenous base comes off of there, right? Here's the 2' carbon, the 3' carbon, that one's really important. 4', and then sticking up like a little antenna is the 5' carbon. And the 5' carbon is connected to the phosphate group. Now that we understand what 5' and 3' refer to, we can get a deeper understanding of why Watson and Crick said that DNA, the DNA molecule, is antiparallel. And that just means that chemically, the two strands of the DNA double helix are running in opposite directions. Kind of like cars going down a two-way street. Right? The lanes are parallel to each other, but the cars are going in different directions. The same is true for DNA. And we name those directions based on the sugars, or the carbon in the sugars that are on either side. So you can see on this side, the 3' carbon, there it is, this DNA strand runs this way from 3 to 5, right? And so here's the 5' carbon right there. All right, and then this other strand goes the opposite direction because it's antiparallel, right? And so you can see this time the 3' carbon is up here and our 5' carbon is down here. And we can also see that the the cytosine and guanine are paired. They're held together by 3 hydrogen bonds, and adenine and thymine are also paired, and they're held together by 2 hydrogen bonds. Now, the way I remember which side is the 5' side and which side is the 3' side is this little trick. the carbon that's next to the phosphate is number 5. So phosphate and 5 both have the f sound, and so it's an easy way to remember which side is the 5' side. And then 3 has that long E sound, and sugar, like deoxyribose, sugar is sweet, and that has 2 E's in it, so that makes that long E sound. So I remember 3 is sweet, and that's the one that's right here on the bottom of the sugar. So now, that gives us all the background we need to understand the details, the process of replication. So replication, here's your double helix. It begins when an enzyme called helicase, it's a protein, comes through and unzips the 2 strands of the double helix. And when it does that, it breaks those hydrogen bonds that are holding the nitrogenous bases together. So helicase, and that makes sense because ACE is an enzyme. Lots of enzymes break things apart. And it's a helicase, so it's breaking the helix. All right, and after those have been broken and those strands are now open with little bases that need partners, an enzyme called DNA polymerase comes in and it adds those new complementary bases. So you can see here on this top part, your A is being paired with a new T. Here the C, DNA polymerase is bringing a new G. And on the other side, DNA polymerase, again, because there's anti-parallel situation, we have to go like this. So this DNA polymerase is adding a T here and a G here and is moving in this direction. So now we're going to look at even more details chemically of this process right here. If you're in Mod 2, this is as far as you need to go. But if you're in AP Bio, let's keep watching. There's a lot going on here, but this would be a great time to use colors in your notes. All right, so when we're adding new nucleotides, when DNA polymerase is adding new complementary bases to our open strands, it has to add them from in the 5' to 3' direction. So, right, it's got to find a little phosphate 5, right, and add our new ones onto that end, building towards the 3' direction. So let's just give a little look here. Okay, so if red is our parent strand, and this is the 3' end of the parent strand, the other side, because it's anti-parallel, this is the 5' side. So our new nucleotides are going to be added from the 5' to 3' end, going this way. And then, right, DNA polymerase just smoothly moving from 5 to 3' to this, which is called the replication fork. All right. Now, on the other hand, right, we still have to go from 5' to 3', but this strand, it's actually called the lagging strand because it's a little more complicated, the situation, so it actually physically goes slower. It lags behind the addition of nucleotides in the leading strand. But what happens is, since there's no 5' anything to add to when you're coming in this direction, what happens is there's a little piece of RNA, an RNA nucleotide called an RNA primer, and I've written these in green on here, and it will attach to one point on the open strand, sort of giving the DNA nucleotides a home base. It gives them a 5' end to build from. So in this little, right here I've got a U primer, and we're adding these purple new nucleotides from 5 to 3. Awesome. And then, so DNA polymerase added those. Then another RNA primer was added, and DNA polymerase kind of jumps and goes a little bit at a time. So it just, instead of continuously, like in the leading strand, replication is happening on the lagging strand, little like chunks at a time. And those chunks have an official name, and it's called Okazaki fragments, after the man who actually discovered this whole leading strand, lagging strand thing. Now, we have a bunch of chunks of DNA now with some RNA sprinkled in, but that's not going to work. So what happens is, after we have these chunks of Okazaki fragments filling in, a protein called DNA ligase comes through and it does a few things. First, it takes out these RNA primers, it gets rid of the RNA, inserts a DNA nucleotide where it belongs. So for instance, this U would be taken out by DNA ligase and a T would be added in, right? And this RNA G would be taken out and add a DNA guanine in there, okay? And then after that, it will actually then go through and zip up and finally put together all the phosphodiester bonds to solidify that final new strand of DNA. And the last thing to talk about is just the fact that it's not like replication happens at the top of a chromosome and you have this huge like zipper unzip and an entirely huge chromosome like unzips all at once. That would take forever. So our chromosomes actually have multiple origins of replication, or places where replication starts. So right here, if this is our parental strand of DNA, there are these little bubbles that actually come up, and this replication bubble will open and replication will start happening in two directions. And so those bubbles will get larger as DNA replication continues to go, and then those bubbles will finally fuse until you have two full chromosomes that have been replicated.